All right, on our next talk, we're going to hear from Ewan McRae at Houston Methodist Research Institute, and provide what I think is, really important and helpful kind of user perspective and sharing their experience with specifically related to challenges of associated with cryo-EM structure determination and modeling of RNA. Ewan, thanks for joining us. Take it away.
Thanks for having me. Can you see my screen okay? Can you hear me?
Sorry, yes, we see it and hear you fine. Great.
Okay, let's start sharing again. Yeah, so first off, I'd like to start by thanking the organizers at PNCC, especially Omar, for inviting me and putting together such an informative workshop. It's been a real pleasure, and I feel like I've learned so much. It's been nice putting faces to a lot of the names who I already recognize through the software that I use to model and validate my RNA structures. Even though I'm just sitting here in my office, it feels like I've taken a step up onto a big stage. So I'm really happy to be here to share with you my perspective on some of the challenges I run into with modeling RNA into these cryo-EM maps. It's also been nice to see several examples of RNA modeling tools and validation tools in a structural world that's often very protein-centric. But for those of you who don't know, if you subscribe to the RNA world hypothesis, RNA has been making these complex structures since the primordial world, way back to when it was acting as this mythical replicator, making copies of itself and starting life as we know it, to when it eventually evolved into this workhorse translator that makes all these proteins that we're so familiar with, and eventually evolved to make these complex regulatory networks mediated by interactions to these proteins that it makes. And now we find ourselves in the modern era where we can sort of design our own RNA structures on the computer, order them through whatever DNA synthesis company we like, make them in the lab, and then characterize their structures and try and get them to do cool and useful things. And one of the ways we do this design is through a method called RNA origami. Very much like paper origami, what we're doing is controlling the folding pathway of RNA to arrive at a 3D structure that we have in mind. And to make these 3D structures, we start with examples of RNA that we find in the structural databases. And we really use this principle of RNA modularity, which goes back quite a number of years, that you can basically take an RNA structure, cut it up, and paste it into a new 3D structure. And it'll still fold in the same way as long as your sequence is correct. And the second principle that we use quite heavily is that a lot of these sequence space is very redundant. So you can essentially program new sequences as long as it has the capability of forming the same secondary structure pattern, and you avoid certain topological traps and potential misfolds that can be introduced during synthesis. So once we've come up with a 3D model and a 2D blueprint, we can use our inverse folding algorithm to generate a sequence. We can then order and transcribe this sequence, do some experiments to validate. This is sort of our workflow. And as I was joining Etta Anderson's lab as a postdoc back in 2019, they were working on RNA origami version 2.0, where we've essentially extended this software to be more automated. We've increased the number of different modules you can plug and play with. We have this nice RNA build program that'll do a lot of the rigid body rotations and assembly of these fragments for you to produce an initial model. And we have some folding analysis software that'll help you avoid some of these topological traps. And we rely heavily on the Vienna RNA package to validate our in silico designs before we go experimental. So one of the things we wanted to do in Eva's lab as we were shifting towards cryo-EM, away from AFM as a primary characterization method, was build into this third dimension, which cryo-EM is so good at looking at. So we can do this by programming this consistent curvature into a series of RNA helices and arrive at something like this 6-helix bundle, which I'm going to be talking to you about as an example today. So here's a blueprint for that 6-helix bundle, and it can essentially be thought of as a series of helical elements. So here in this top helix, we have 1, 2, 3, 4, 5, 6, 7 different helical elements that we can think of, and they all form a coaxial stack to make this elongated rod that we see in many of our structures. We can feed that blueprint into our design algorithm and get a sequence, and we validate the structure with the RNA. We can predict that it'll still fold this, and then we go to our cryo-EM workflow. So as I mentioned, we purchase the DNA and we make everything in vitro. So we're very confident the sequence we're looking at is the sequence that we think we're looking at. So we don't have this problem of identifying sequences in our maps. We essentially just assume that we have the right sequence. And still the most important step in this workflow is the sample purification. If you can get a nice pure RNA sample that's well folded and you can get nice dispersed particles on your grids, a lot of the data processing really does itself, does its own work. And then we get to the model building. Unfortunately, what we often find, especially with these larger RNA origami structures, is that we end up with low resolution maps compared to protein structures. And when I say large RNA origami, I'm talking about 700 nucleotide or even longer single strands of RNA. And there's a few reasons why we end up with these lower resolution maps. And one of them is that we're throwing away a lot of data during 3D classification. So when we pick particles initially from our micrographs, we might end up with something like this that gives us a reconstruction of a 6-helix bundle. And then we'll do sorting of these particles into different classes in 3D. And we isolate the particles that give us the highest resolution reconstruction. And we're essentially throwing away a bunch of data that is presumably looking sort of like this, at least more so than our noise classes or other classes. And so what we'd really like to be able to do is incorporate all of that data into our model building and our map reconstruction. So what we think is happening is we have this design of a nice single structure. We make it, and it actually looks more like this. It's sampling a whole bunch of different conformations in this continuous landscape. And then we select for the highest resolving particles. Now there's a bunch of tools that have come out over the past 3 to 5 years that have this promise of solving these motion trajectories and being able to use the signal from all these particles and all these different conformations to reconstruct an even higher resolution consensus structure. And there's some examples that these work for proteins, but unfortunately we haven't seen any of that higher resolution information coming out when we look at our RNA only structures, maybe because of a lack of data in the training sets for these models. But what we often get from these sorts of analyses are, whoops, movie's not starting. Realistic looking motion trajectories. So here's a video of this 6-helix bundle. If we do principal component analysis to sort particles along different components, we can see these sort of wiggling or breathing motions in most of the trajectories, which we think represents some of the dynamics in our structure. But what we'd like to be able to do is model each of these conformations more explicitly. One way of looking at all the data and not discarding anything is this non-averaging approach. And this spurred from a collaboration with Gary Wren and his scientist Yan Fang at the Molecular Foundry in Berkeley, where they use this IPET technique to reconstruct individual particles without averaging. So we sent them some of our 6-helix bundle sample and they imaged it for us. And after doing some denoising, you can reconstruct individual particles. But then we have our first problem of validation, and that's just validating the resolution of the map we get. Because we only have a single tilt series for our reconstruction, we don't really have independent half sets that we can use to calculate this gold standard Fourier shell correlation and get an accurate estimation of our resolution. If we split the tilt series, we get an overestimation of the resolution, presumably because the half sets share a certain amount of noise. We can do things like compare to our single particle averaging maps. If we pick a map from our individual particle reconstructions that looks very similar to our single particle averaging maps, we can do this for a shell correlation, see a resolution range 25 to 20 angstrom. But again, we're sort of picking what we're comparing here. What we're really interested in are the individual reconstructions that look different than our single particle analysis reconstructions, which we should zoom out to see soon. So we, or Yanfang, reconstructed 120 of these individual particles, and we performed rigid body fitting of these 7 helical elements in each of these rods in Chimera X in an automated way. So we're not getting maybe the most perfect models in terms of clash score or preferred angles, but they're a rough approximation of what these structures might look like. And we can do this for all of these until we have 120 rigid body fit models. And then we can sort them along basically a trajectory based on certain interhelical angles. And when we do that, we call this a maturation percentage. Oh, sorry. My phone is talking to me here. Sorry about that. We can come up with a prediction of how this particle might change over time. But how do we validate the resolution? Perhaps a better way than these Fourier shell correlations is looking at the features that are actually discernible in a given map. And there's a couple ways we can do that. We can again look at our PDB model that we built into our single particle map, and we can look at our single particle map filtered to different resolutions and compare these to the maps we get through the IPET method from individual particles. And we can look at their side views and top views and a cross section, and we can compare the features that we see. So we know that each of these helices is about 2 nanometers in diameter. And so their centers should be about 2 nanometers apart. And so if we can resolve 2 helices that are right next to each other, we might say we have close to this 20 angstrom resolution. And how do we validate the models? Well, we use these models to generate a hypothesis, and then we try and test that hypothesis in vitro back in the wet lab. So as I mentioned, we sorted these particles along this maturation percentage trajectory, where it encounters reconstructions we have through single particle methods. And we see that we predict that it gets more and more compact over time. And so we looked at this sample freshly transcribed by X-ray scattering in solution, and we saw over a 17-hour time course that we do see it get more and more compacted over time. And if we look at the initial 15-minute data window to the last 15-minute data window from that SACS data set, we can see that there's a good correlation to our models from the early and late maturation percentage models. And we can see there's a nice transition that seems to happen after 7 hours. So we're following this very slow maturation process. We can go further than that and make a fresh sample again, plunge freeze it immediately after purification, and we can reconstruct something that looks like this early conformation. If we leave it out on the bench for about 18 hours, we can get a data set that has entirely this mature conformation, as far as we can tell from our analysis. And if we plunge it at an intermediate time point, we can get a combination of both of these structures in a single data set, where this one doesn't quite look as mature as the fully matured one, but still has a little kink in this final kissing loop. Yeah, so some of the challenges here is distinguishing between this continuous heterogeneity, this RNA wiggling or breathing that seems to happen more so than it does in proteins, this rugged energy landscape of RNA. Also, certain deep learning tricks, especially that work for proteins, don't quite work for RNA yet. So that's on our wish list, is to have more of these tools that are RNA centric. And for our lab, at least, we seem to hit this resolution barrier right around 5 angstrom, where it doesn't matter how many more particles, how many more movies we collect and particles we pick, it's really hard to get past this 5 angstrom limit. Okay, so another challenge with modeling RNA is that unlike proteins, where you have these sort of 2 backbone angles that are fairly well described, and you can fit them on a Ramachandran plot and find outliers pretty easily, RNA backbones are described by 6 torsional angles, as well as rotation around the glycosidic bond. So you have a lot more parameters to play with, which means you can overfit models quite easily. Yeah, so another important aspect of RNA structure are these non-canonical structural features, which show up quite a lot. Whereas most people are taught about these 4 base pairs, AU and GC base pairs with these Watson-Crick faces, there are actually 192 possible different base pairs that can happen, and most of them have been observed. And this is because not only do you have this Watson-Crick edge of a base, you have the Hoogsteen edge, and you have this sugar edge, which are capable of hydrogen bonding. And you can have different orientations of the backbone. They can either be cis or trans. And so if you think of a base as this triangle with 3 edges, there's 6 different ways you can put it together, and then you can have it in cis or trans. And then for every possible base combination, you get up to 192 possible base pairs. And these are really hard to predict. A lot of current tools don't have good weights or restraints, even for things that are fairly common like GU bases. And we see these in really important regions of our structure. So here's an example in one of our RNA origamis where we have this central kissing loop motif, where we have 6 unpaired adenine residues. At least that was what was determined in a crystal structure. But in our cryo-EM map, we actually see fairly decent resolution for this trans Watson-Crick A to A base pair. If we really push the contour, we can see base density. But the rest of the map is fairly low resolution. Again, right around this 5 angstrom barrier. So how can we validate that this is the correct structure modeled into this map? Well, one way is to compare it to other models. So here's the X-ray structure model that we used to make our initial design. And you can see it doesn't fit very nicely, but also it's more extended if you measure the distance of a full helical turn. It's about 3 angstroms longer than our model, which is about equivalent to 1 base pair step. And actually what we had been observing in our original designs is this twist that sort of propagates through our structure. And we use these double crossover motifs to keep our helices parallel, but they rely on the underlying assumption that in between double crossover motifs, you have an integer number of helical turns. And because we have this central kissing loop motif that we extracted from a crystal structure, we were modeling it as 9 base pairs, where in our cryo-EM map, it's closer to 8 base pairs. And so this throws off the helical register, and we get this twist that propagates through our structure. So by implementing this twist correction, simply adding another base pair in front of each of these kissing loops, we get a new sequence, and we can get a new cryo-EM map. And we see that this twist is more or less gone, and the through axis helical view, the cross section here, is very close to our design view. So we've validated our model in that way in the wet lab by making this hypothesis and then testing it. Now moving on to some work I'm doing now in my lab, we're really interested in how modified bases affect the structure of RNA. Modified bases have seen a lot of attention recently for their use in RNA therapeutics, specifically 5-methylcytidine and N1-methyl pseudouridine are used in mRNA vaccines, amongst other things. And there's this question, if you start incorporating these modifications, are you going to change the structure of RNA? Are certain regulatory motifs that you want to put into a UTR still going to work? And so this is a question we're trying to answer, first by looking at our highly structured RNA origami designs. So we made this 6-helix bundle that I've been showing you so far today, entirely with 5-methylcytidine, and we allowed it to sit on the bench for 48 hours. And when we reconstruct the monomer, we see that it's still in this young conformation. So we haven't been able to observe this maturation into the more compact structure, which is interesting. And another thing we observed are these dimeric conformations, which we hadn't previously seen with the regular NTPs. And these are quite interesting. We see them with N1-methyl pseudouridine and with 5-methylcytidine. This type 2 dimer is fairly easy to understand. It's these central kissing loops. Instead of kissing with each other, they're kissing intermolecularly. And these barrels just sort of come together like so. But this type 1 dimer is a bit more puzzling. If you look at it for long enough, you can work out what we think has gone wrong, which is misstacking at this 4-way junction here. That leads to this helix 5 kissing loop pointing outwards instead of inwards. And what we think is happening here is a zoomed in diagram of the different helical stacks. As the RNA is transcribed, we have this kissing loop half A transcribed first, then B is transcribed second, and they can fold much faster than the rate of transcription. So presumably these helices are already folded by the time the polymerase is moving on to C. And so they have the opportunity to base stack on each other. And because there are 2 cytosines at this junction with a methyl group now, this stacking interaction could be stabilized. And so they could sample this initial alternate stack for longer than the NTP transcribed structure. And this allows them more time to bump into another misstacked bundle and form these kissing loop interactions intermolecularly, which are then quite stable and essentially irreversible. So we have this 2D model that explains this to us and allows us to test the hypothesis that cytosines at the junction are what's causing this dimer formation. So with size exclusion chromatography, we can actually separate the dimer and monomer and some abortive transcripts. And we see with NTPs in red, we have essentially a lower amount of dimer. But when we transcribe with 5MC, we have quite a high amount of dimer, even more than we have monomer. But when we junction correct and remove any of the cytosines from the junction, we have similar amounts of dimer to the NTP transcribed sample. So again, we have this hypothesis generated by our, in this case, 2D model that we're validating in vitro. Now, reviewers want us to build models for these anyways. We're not quite sure what the utility of a 12 angstrom RNA model is in the PDB. Maybe other people here can comment on that, but we're happy to oblige. So here I'm going to show you some of the modeling workflow, which is pretty much the same for higher resolution maps as it is for this low resolution map. We start with a model either from RNA build or in this case from a prior PDB. And we use Isolda and Chimera X to get most of the fitting right. For these first 4 helices, they haven't changed much. The real change in the structure is from, helices 6 and 5. So we can then, this is replaying. go and get these rigid helices and plunk them in to our map, like so. And then what we do is we use a program called Drafter to basically fill in all the gaps, refine around the junctions that are sort of sloppily placed together with the rigid body fitting. And Drafter, if you allow it to do several thousand models, you can pick the top scoring 10, calculate an RMSD between them, and sort of pick the best model and go from there. And this usually gives us a nice place to work from. So we can model with Drafter this new alternate stack and extended kissing loop, at least for this monomer. And this particle is quite symmetric. Actually, we've enforced C2 symmetry on this map to help modeling a little bit. But when we flip the second unit round, you see there's quite a lot of clashing here on this intermolecular kissing loop. So again, we just remodel that with Drafter. It gives us something reasonable looking. And then we can stitch them together with some PDB tools. And we now have this complete structure built through a combination of Chimera X, Isolda, Drafter, that looks reasonable with a fairly high clash score. So what we use to get that down is this program called QRNAS, Quick Refinement of Nucleic Acid Structures. It's actually not super quick once you get up to trying to optimize 1,400 base pairs at a time. This run goes overnight before we get through 5,000 iterations, but it does a good job of reducing the number of clashes predicted by Chimera X. And then we'll take this to Phoenix and do a validation. And you can see it's quite bad. Phoenix doesn't like a lot of the bond angles. They're far outside the range, but it's got a good mole probity score and a good clash score. Phoenix also doesn't like that we have no favored Ramachandran angles because we don't have any protein. So Phoenix would like us to have some protein in there. And the model to data fit isn't great either. And here we are sort of at this dilemma that was talked about by previous speakers, where how much do we refine a low resolution model? Which metrics do we focus on? Do we want to try and optimize bond angles and fit to data, or are we happy with these good looking mole probity and clash scores? If we do some Phoenix real space refinement with base pair restraints, quite heavily restrained, I should say, the angles get a lot better according to Phoenix, but the clash score is quite bad, I would say. But the fit to the model is, the fit to the data is better. But if you actually compare these in Chimera X, you can hardly see any difference. So it feels a bit futile to try and keep refining this. So here are the tools we use for RNA origami model building. We generate these ideal helices or use the PDB for other more complicated motifs. And we sort of plunk them together in Chimera X, morph them with Isolda. And then we run this iterative loop of Phoenix real space refinement and QRNAS until we end up at a model that we're happy with. And then as a final step, we usually go through COOT base by base to fix any weirdness. Sometimes we find a different sugar pucker that looks better than another one, so on and so on. Okay, and the final example I'm going to talk to you about today is this not RNA origami structure. This project was a collaboration with Phil Holliger's lab at the LMB in Cambridge, just at the end of my postdoc in Ebba's lab. And his lab is interested in this RNA world hypothesis and evolving these RNA enzymes that can make copies of themselves. So these RNA polymerase enzymes. And they started with the sequence based on this RNA ligase enzyme, whose structure was solved in 2009, a nice crystal structure. They started with this as their sequence, and they perform in vitro evolution experiments where they have mutants with extended 5' and 3' regions, and they try and improve the activity of this thing and make it more polymerase-like and less ligase-like. And what they ended up at was this interesting heterodimer, where when they tested the activity of their most enriched sequence, this 5TU, it didn't have as good activity as the pool of RNA did. But when they combined it with the second most enriched sequence, it has enhanced activity. So it's functioning as a heterodimer, and somehow this T1 is enhancing the activity of TU. And T1 has no activity on its own. So this is a pretty interesting problem. We tried to tackle it with cryo-EM, and there was a really fun model building step. So I'm going to talk to you a bit about that. So here we have the predicted structures of 5TU and T1 and their sequences. You can see they share the same core sequence, but they have different 5' and 3' modes. We were able to get a model that has the right size to be a heterodimer. Again, we got pretty much hard stuck at this 5 angstrom limit. Collecting 20,000 movies, we weren't able to push past it. And we already got to 5 angstrom after like 8,000 movies. So it seems to be a hard limit for a lot of our stuff. So we start modeling the unknown is a bit more challenging than modeling RNA origami because we don't have an initial map to go on really. With this 5TU, we know it's active, so we know it should look something like the progenitor ligase enzyme. So we can actually model the crystal structure into our map, which confirms our initial secondary structure guess is more or less correct. If we look at the secondary structure prediction from Vienna RNA, it's actually quite poor. It predicts different hairpins, and if we use this as an initial starting structure, we wouldn't get very far with modeling this. But we can use these secondary structure restraints in Drafter. Even amongst the top 10 models, not all of them have the correct helix placement, but the best one does. So we go with the best one and we continue modeling from there. Again, we use Drafter to fill in the gaps at these new junctions and regions that aren't in the crystal structure, maybe aren't as well modeled. And we generate a larger number of models, and we get a better pairwise RMSD between them, and we're more confident that these are accurate. Then we do our usual rounds of Isolde, Phoenix Real Space Refined, and QRNAS to get our final model, where we can see some nice features popping out that are similar to the crystal structure, and so on and so on. And then it comes to modeling the other half of this heterodimer, this T1 factor. When we initially tried using Drafter with this secondary structure prediction, it wasn't succeeding in finding a model that fit to our density. But when we used the Vienna RNA secondary structure prediction, quite quickly it converged on this alternate structure that I will show you here. And we tried to validate this alternate new structure by doing this deep mutational scanning, where we ordered a whole bunch of different variants of T1 and 5TU, and we did the in vitro selection experiments and observed which sequences were enriched and not enriched in the selection, and used that to validate some of these base pairs, where if you have compensatory mutations, the enzyme still works, but if you have disrupting mutations, the enzyme doesn't work anymore. And once again, we iterate this through Isolde and Phoenix Real Space Refine and QRNAS to get our idealized model with the secondary structure. And then we put together the interconnecting motifs, again, modeling them individually, first with Drafter, and then through our iterations of Phoenix and QRNAS. And from this structure, we can generate more hypotheses about how 5TU works and why T1 enhances its activity. So to just summarize here, tools for things with loops, so not RNA origami. Again, we use RNA helix from Rosetta to generate helical fragments, and then we have to guess at the secondary structure. And sometimes it takes a couple of tries, and there's a couple of different structure prediction tools, so far it's worked out. And then we like to validate the experiments. I hope you recognize that was a sort of common theme in our validation of models built into low resolution maps. We use Drafter and semi-auto Drafter to place helices and model loop regions, and then this iteration that I've mentioned a few times now. The challenges, low resolution model building is tough. There's many possible structures that can fit within the map, and we're just showing one that we think is the best. So there's quite a bit of bias introduced there. And as we saw earlier in my slides, RNA is pretty dynamic. Likely it's sampling a lot of very similar local conformations. We have a certain amount of knowledge about RNA structure in general, from structure predictions informing base pairing and physical parameters informing the backbone geometry, which allow us to make these models at higher detail than our maps are. And again, we have to validate these by returning to the wet lab hypothesis generated from our models. And then there's the conundrum of how do we like to improve our models? Is it towards lowering the clash score in the backbone or towards fitting our map better, getting higher CC and Q scores? Probably the former for low resolution maps would be my opinion there. And I'd just like to acknowledge collaborators in the Anderson lab, as well as Cody Geary and Jan Scott Pedersen, Gary Wren, who helped with the RNA origami design, SACs, and individual particle tomography, respectively. CryoEM facilities, where I collected the data I showed you today. Of course, the PNCC, where I hope to collect some data soon. Funding sources were funded by the Cancer Prevention Research Institute of Texas and Pew Charitable Trust currently, and postdoc funding from NSERC and the DFF. And I'd also like to thank everyone in my new lab here in Houston for making every day exciting. And thank you for your attention. I'm happy to take any questions.
Ewan, thanks. That was great. And yeah, I think you gave us all a good sense of how difficult working on these projects can be. Happy to feed you some of the questions. This first one is a little bit long. I might try to paraphrase, but I'll leave it up if there's some more detail that you wanted to go back and address. It's related to questions on your observations of the overall kind of conformational dynamics that you're seeing in the RNA molecules. And this person is particularly interested in how this might reflect kind of exposing cryptic sites for small molecule drugs. And if you might be able to comment on RNA cryptic sites and drug discovery and conformational states they exhibit.
Yeah, there's definitely a lot of opportunities for small molecules to sort of intercalate in between bases because of that, breathing and opening up and closing that we think is happening at a time scale like that. So as far as modeling it goes and drug design, I know there's lots of people who are trying these sorts of approaches. Unfortunately, I still think that machine learning based RNA structure prediction and ligand docking is pretty far behind protein stuff. And we've seen earlier the protein stuff is also still ambiguous with docking these small molecules. So I hope that answers that.
I might kind of extend this a little bit to, I'm a protein-centric person, and so I don't follow the RNA field as closely, but we all kind of get wowed by these really high-resolution ribosome structures. What do you think the difference is there? Is it because of the integration of the proteins, or is the ribosome kind of its own beast compared to other natural, or in your case, unnatural RNA structures?
It's probably a combination of those things. I mean, the ribosome has had billions of years to evolve into a highly structured thing, so it's probably fairly optimized in terms of its structure. It also, you don't want to be degrading ribosomes all the time in your cell, so it's probably minimizing how floppy it is and how many conformations it can adopt that'll lead to its destruction, right? So also the size probably helps a lot with ribosomes. These things are, bigger than your average RNA structure, but still relatively small. We're talking 300 kilodaltons versus a few megadaltons for a ribosome.
Are you guys seeing any in your designs? Do you see correlation with topology or size or anything like that that relates to the conformational dynamics or resolvability that some empirical kind of observations from your lab?
We see some things related to topology. So we have one structure called the PXT. It sort of forms this paranemic triangle, and it's quite rigidly locked in there just because it's so tight and constrained. So we often get pretty good resolution of the core, but at the helical termini, we still see a lot of that moving around. So
Sure.
I think, yeah, it's, and then for our bigger samples, as they get longer, they can, they have more twist for all. They are getting to that length where
Yeah.
they're past their persistence length.
Yeah, that makes a lot of sense. Let's see here. So one of the audience members asked if it's possible to incorporate NMR restraints. I might just extend it to other experimental restraints into EM refinement for RNAs.
Yeah, actually, we've had some folks do that for us and with really good results to improve the metrics, validation metrics for our models. We haven't gotten around to getting that software installed locally to be able to do that for all of our models, but we probably should. Yeah. Again, it's sort of a, how good should we make the model for our 7 angstrom structure or the PDB?
Yeah, no, it's a tough
That's what it
question.
comes down to.
I think with not only for RNA or for anything at those more modest resolution limits.
If it comes easily, I'm happy to make it as good as possible, but
right. Let's see. Do you think it is possible to see the different conformations between RNA synonymous mutations by cryo-EM? I'm not sure about synonymous mutations, but
I think they mean like if you have a GC base pair and you switch it to GU or
thanks.
something like this. It's tough because we often don't have the resolution to see individual bases, let alone the chemical difference between individual bases. Like I showed on one slide where we have this AA base pair. We only know it's AA because we have a good idea of what the exact sequence is, and that's what fits in our model. We can't discern bases at our resolution. Yeah.
Yeah. All right. And then another question, I think kind of related to what we were talking about just a minute ago, but they're specifically asking about structures of some ribozymes, some relatively low molecular weight ribozymes that have been resolved to closer to 3 angstroms. For example, the tetrahymena ribozyme at 3.1. And do you have any comments on, why some even smaller functional RNAs might resolve to such higher resolution?
Yeah. Yeah, so that's a great example of a nice high resolving RNA structure. And it's sort of like the poster child for RNA structural biology. And I think the difference between that and our RNA enzyme is that ours is evolved in vitro. And again, that one's a natural RNA. So maybe it's just a feature of naturally evolved RNAs that they can get to more stable structures than the ones we design or evolve in vitro.
Thanks. I could jump in and ask a question real quick.
Yeah, please.
You had mentioned the fear of overfitting because of the number of parameters and because the resolutions you're working with. Are there geometric hallmarks of that? Like, do you start to lose planarity of bases? And what kinds of restraints are you applying to mitigate that?
Yeah, that is what we observe actually. If we over-optimize the bond angles, we get non-planar bases. And then we go back and fix that with qRNAs. It's quite stringent for enforcing base planarity, but then our bond angles get worse. So yeah.
So I'll add a question too that I was struck by. I really like the attempt to characterize the conformational heterogeneity using single particle tomography. I think, for, being able to leverage the high signal to noise from RNA to get nice signal from those types of data. And I might have missed it, but were you able to capture unique conformations as compared to what you're able to get from like 3D variability analysis that you showed on your standard single particle workflow?
Yeah, so we see some conformations that are outside of what we observe with single particle analysis. Yeah, we think they're real. We think they're early in this maturation process. So we have two conformational variability things going on with the 6 helix bundle. We have this maturation process where it gets more compact and has a rotation and a breaking of a kissing loop, a bunch of stuff I didn't get into. And then we have this local continuous conformational variability as well. And so the variability analysis we do with our single particle averaging maps captures this local stuff. And then the IPET has seemingly captured some stuff outside of those ranges even. Again, it's hard to validate. There's a lot of good arguments that it could be electron-induced damage causing weird conformations and stuff like this, but
Yeah, no, I can see that, and also just limitations on comparing maps at such low resolution is challenging.
Yeah.
All right, well, I think we've covered the questions. I'll thank you again for a great talk and highlighting all the challenges in the field and appreciating. Well, I think what I got struck by is just how much effort you have to go through to bootstrap your way into a good starting model. That's that's a lot of work.
Yeah, I think
Yeah.
there are a couple more automated tools even that were mentioned in this workshop that we're going to go back and try with our new models, but I don't think we're quite at the resolution where it'll work well for us, but we'll see.
Great.
But yeah, thanks for having me. It's been my pleasure.
Great.